everyone. Today we're going to propose a bronchial implant for COPD treatment. My name is Emily, and my teammates are Desiree, Marissa, and Mia. Imagine having to gasp for air every time you take a breath. That is what it feels like to have chronic obstructive pulmonary disease, or COPD. COPD is a combination of lung diseases like emphysema and bronchitis that causes airflow limitation. COPD causes narrowing of the airways and causes the bronchial tubes and alveoli to fill with mucus, blocking airflow to and from the lungs. Specifically, the lungs' epithelial tissue secretes mucin proteins that thicken mucus and have been found to unfold improperly in the lungs. If the mucin proteins do not unfold completely, the mucus gets thick and sticks to the epithelial cells. This trapped mucus can cause bacterial growth, which causes inflammation and lung damage. COPD affects more than 15 million Americans and causes 150,000 deaths each year. The total life expectancy loss for a person diagnosed with severe COPD is approximately 8 to 9 years. Having COPD is also very expensive between hospital bills and treatments. According to one study done in Sweden, the annual cost for very severe COPD patients is approximately 17,000 euros or 19,000 US dollars. Furthermore, there is no cure. However, there are treatments. The first and most common treatment for COPD is the use of bronchodilators and corticosteroid inhalers. These treatments reduce symptoms by reducing mucus production and airway dilation. The disadvantages of this treatment are that patients do not always take their inhaler. The inhalers are more likely to cause pneumonia, and it does not stop progression of COPD. The cost can be anywhere from $42 per month for short-term treatments like ProAir to $220 per month for long-term treatments like Spiriva. Another treatment option is a lung transplant, which can improve the length and quality of life. However, there is the possibility of rejection and infection. Some patients are not eligible. The wait time can be up to 2 years, and the cost can be up to $165,000 if not covered by insurance. Another treatment used for bronchogenic carcinoma patients is a hybrid polymer and metal stent that is placed in the bronchial tubes. These hybrid stents help minimize tissue ingrowth and can be easily manipulated or moved around. However, some limitations are that the stent does not remove mucus, can migrate, and can be difficult to remove. Furthermore, the flexible bronchoscopy procedure that is required to implant these stents are very expensive at over $20,000. The image on the left is Boston Scientific's Polyflex stent. On the right is the stent by Merit Medical Systems. Notice the shape of the metal that makes this implant difficult to remove. Therefore, there is a need for an artificial bronchial implant that can help COPD patients live longer and more comfortably with their disease by allowing increased airflow to and from the lungs. The goal of our device is to alleviate COPD patient symptoms by targeting the thickening and overproduction of mucus and inflammation to ultimately increase the airflow without impeding vital gas exchange and immune functions. This device must also avoid fibrotic encapsulation, and the target audience will be mid to severe COPD patients.
Before designing our product, we defined functional requirements. Because the implant is intended for long-term use, the device must strategically degrade and avoid migration. It should also avoid irritating the lungs and become fibrotically encapsulated. Fibrotic encapsulation would be problematic as it would impede the delivery system's functions and make removal way more difficult. Smart polymer properties will also be necessary to allow expansion during device implantation. Furthermore, the implant must be flexible, resist cyclic compression, and be visible on MRIs and X-rays. The team also developed design requirements. Based on previous studies, the device must break down 3,000 USP units of lipase per day in order to properly and effectively break down mucus. The original stent will expand to around 12.6 millimeters out of diameter, which is the average size of a male's left bronchi. However, when the device goes to the market, it will expand to multiple fits based on the specific patient, as fit is a main contributor to anti-migration tactics. The stent must be able to handle a yield stress of 16 kilopascals and function between 8 and 29 pascals of air pressure based on the function of the lungs. Finally, the implant must have an expansion force of 12 to 15 newtons and a flexibility force of 3 to 4 newtons. Ultimately, the drug delivery system should last for a minimum of 2 years. Our device must also be biocompatible and have low toxicity. To be both safe and effective, the implant must release 3,000 USP units of lipase per day and 2.78 micrograms of mitomycin per day, based on literature. The device must also release minimal nickel ions from the nominal core as to never exceed the toxic blood serum level of greater than 4.8 micromolar. To be effective, the device cannot also instigate a detrimental immune or inflammatory response and must avoid fibrotic encapsulation.
To achieve these goals, we have developed a drug-eluting stent that can be implemented into either the primary or secondary bronchial tubes. The device has several components such as a biodegradable exterior that releases lipase and mitomycin, a nitinol mesh interior, pores, and laminin domains. During inhalation, air passes through the trachea into primary bronchi, the secondary bronchi, and then into the alveoli, where exchange of oxygen occurs. During exhalation, carbon dioxide exchanges through the alveoli walls and back through the bronchi and trachea and out the trachea. Our device will be implemented into the primary and secondary bronchial tubes to help assist with airflow. The stent's geometry will be patient-specific based on MRI analysis. In general, the device will be around 13 millimeters in outer diameter, 1 millimeter thick, and 0.5 centimeter long. This is a drawing of our device. The nitinol mesh is embedded in a polymer shell to mitigate migration through mechanical loading on the surrounding endothelial tissue. The nitinol is mechanically polished to increase corrosion resistance. The coil shape is set at body temperature and can be manipulated at room temperature for easier insertion and subsequent expansion. Nitinol has been proven biocompatible, being used in several airway stents currently on the market. Poly-L-lactide is used extensively for bioabsorbable stents. As the PLLA degrades, it will release lipase and mitomycin. The elasticity allows for slight movement during nitinol expansion. These properties can be adjusted by optimizing the temperature and duration of ring opening process during fabrication. The pores running parallel to the nitinol coil are either formed during PLLA fabrication or laser cut to allow for cilia and immune cell protrusion and movement. There are also 2 laminin regions to promote the adhesion of endothelial cells, anchoring it in place. The enzyme lipase will break down mucus plaques and mitomycin, preventing hyperplasia. Dosages are determined based on the release rates previously described: 1,102 milligrams of lipase and 36 milligrams of mitomycin, which are released over 3 to 4 years. Nanoparticle encapsulation of mitomycin by a polyester allows for different release rates. This is a cost breakdown of our device. Our device costs $3,656, which is comparable to our competition. The device is sterilized using electron beam and then implanted during a bronchoscopy. It expands at body temperature, and the laminin regions increase stability. These regions are very thin, and the rest of the device releases mitomycin to reduce hyperplasia and limit the amount of tissue damage sustained during removal. The device will be removed after 3 to 4 years once it becomes inactive to prevent fracture, which could increase nickel ion release, and to mitigate inflammation and thrombosis from prolonged implantation. In summary, our device is different from alternative treatments because it physically opens the airway while targeting the overproduction and accumulated mucus plaques. The stent also utilizes strategic cell adhesion and expansion to prevent migration. Ultimately, it alleviates symptoms and allows for vital lung function to occur uninhibited.
We will perform various tests to assure our device meets the aforementioned functional and design requirements. These benchmark tests are crucial to assess preclinical efficiency and are necessary before moving on ethically to animal testing. To assure our device produces a satisfactory drug release profile and releases the 3,000 USP units of lipase per day, we will perform a dissolution test with a flow-through cell apparatus. The airflow through the device is another major component that needs to be tested. Previous studies have outlined the creation of a pneumatic device to stimulate breathing on a cellular level, through which we could compare the air pressure of a bronchial model both with and without our implant. In order to meet the ISO standard 10993, we will perform various biocompatibility assays. First, to assess any cytotoxic effects, we would use a colony formation assay with the V79 cell line. We would also look at the implant's potential to cause cell lysis through layering the device material on mouse L929 fibroblast cells. We would use scanning electron microscopy to assess thrombus formation on blood exposed to the material, a crucial aspect in determining hemocompatibility. To determine if the device might induce a detrimental amount of inflammation, we will run an immunoassay for TNF alpha, which is a cytokine mainly produced by activated macrophages, T lymphocytes, and NK cells. Carcinogenity will also be tested in vitro through a mouse lymphoma TK assay. In all of these tests, we'd be using a non-drug eluting PLA and nitinol stent as the control material to compare the experimental results to. We'd also measure the device's impact on wound healing and tissue interactions. We would use an ELISA assay to measure the chemokines released from platelets to assess the level of platelet activation on our device while compared to the control material. Lastly, we would use fibroblast media to study nickel ion release levels before moving to in vivo testing. ASTM F981 requires multiple species to be used in preclinical animal tests before moving on to human testing. First, we plan to test our device in a small animal study of 25 mongrel or mixed breed dogs, as they have been proven to be a successful model in the study of drug-eluting airway bypass stents. Our stents will be placed in the dog's primary bronchi, since this standard requires the operative site in the chronic animal study to be similar to the device's intended application in humans. This will also allow our device to contact pulmonary tissue. Ideally, the study would run for as long or longer than our device would be implanted in humans, so we would aim for a 5-year-long study. For control measure, PLA stents that do not elute drugs would be used and implanted into a portion of the study dogs to compare to. We would use this study to first assess the clinical efficacy of our device and its ability to perform the desired functions. We would also assess any systematic or acute toxicity, mucous membrane irritation, fibrotic encapsulation, and degradation during the long-term implantation, and also through examination of the device after explantation. We will study pigs as our large animal model, as pigs have been previously used to study tracheobronchial stents and have a similar bronchi diameter to humans. Our stents would also be placed in the pig's bronchi for 5 years to assess the clinical efficacy and the same device safety parameters. We would use the non-drug eluting PLA polymer once more as a control measure, since ASTM F981 requires the use of the same control material in both the chronic animal demonstration tests and any acute testing. However, these limitations to our testing include in the cytotoxicity test, uncontrolled physical interactions can have an impact on cell lysis measurements. Also, thrombin can confound platelet activation in our hemocompatibility assay. And living system testing will be limited by the anatomical differences between the testing species and humans, specifically the differences in pulmonary and immune systems between the two species. However, this testing is still crucial in proving our device's safety and effectiveness.
In conclusion, the bronchial stent will be the first stent for COPD. It will break down mucus, enable easier breathing, and increase COPD survival rates. It is better than the competition because the patient will not need to worry about the treatment compliance with inhalers and will be able to receive this stent more easily than a lung transplant. However, there are still some limitations to this option. The bronchial stent will use nitinol, which has the potential to release toxic nickel ions. It will be implemented in a sensitive area of the body. It will require replacement after 3 to 4 years, and it uses PLLA, which degrades by bulk degradation. Therefore, once the interior is completely degraded, there's going to be a burst release of the drugs as the structure collapses. PLLA also produces lactic acid when degrading, which increases the acidity of tissues around it. While it has these limitations, it's still used extensively in cardiac stent, and that's why we chose to pursue nitinol as use. In the future, composites, nanoparticle encapsulation, or surface coatings can be explored to mitigate these limitations. Overall, the cost of this device is estimated to be $3,656, which is comparable to other methods. Here are our references, and we look forward to your questions.